This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to our exploration of one of nature's most remarkable success stories. Over the past 200 million years, mammals have evolved from tiny, shrew-like creatures scurrying beneath dinosaurs' feet into the incredible diversity we see today, from blue whales to bats, from elephants to humans. In this narration, we'll journey through deep time, discovering how our mammalian ancestors survived mass extinctions, adapted to changing climates, and ultimately came to dominate virtually every habitat on Earth. Prepare to uncover the fascinating story of our own evolutionary family tree. Chapter 1: The Synapsid Legacy. The story of mammals begins not with furry creatures nursing their young, but with animals that would have looked quite alien to our modern eyes. If you could travel back to the Carboniferous period, around 320 million years ago, you would witness a pivotal moment in the history of life on Earth. The great family tree of land-dwelling backboned animals was splitting into two fundamentally different branches. One branch would eventually give rise to lizards, snakes, crocodiles, dinosaurs, and birds. The other branch, known as the synapsids, would lead to every mammal that has ever lived, including you. The distinguishing feature of these early synapsids was deceptively simple: a single opening in the skull behind each eye. This temporal fenestra served multiple purposes. It lightened the skull without sacrificing strength, conserved energy by requiring less bone to build, and provided attachment points for jaw muscles positioned farther from the jaw itself. This arrangement allowed the muscles to be longer and generate more powerful bites over a wider range of movement. Meanwhile, the other branch of amniotes, the sauropsids, had a different skull configuration, with two openings on each side. These anatomical differences might seem trivial, but they set the stage for dramatically divergent evolutionary paths. The synapsids would come to dominate terrestrial ecosystems for tens of millions of years, long before the first dinosaur ever drew breath. The earliest successful synapsids belong to a group called palicosaurs, though this term has fallen somewhat out of favor among scientists who now prefer stem mammals or basal synapsids. These animals flourished during the early Permian period, roughly 300 million years ago. Among them was Dimetrodon, a creature that has confused generations of schoolchildren who assumed it was a dinosaur. With its distinctive sail-like back and lizard-like stance, Dimetrodon looked nothing like any modern mammal. Yet this apex predator, stretching over 4 meters in length, was actually more closely related to you than to any dinosaur that would come later. The palicosaurs displayed remarkable diversity. Cotylarhynchus, another member of this group, presented a radically different body plan. This massive animal reached 6 meters in length, possessing a barrel-shaped body mounted on sprawling legs, topped with a disproportionately tiny head. Its limbs extended sideways from its body, like those of modern reptiles. Yet the single opening behind each eye marked it unmistakably as a synapsid, a member of the mammal lineage. As the Permian period progressed, the palicosaurs gave way to a more advanced group of synapsids called therapsids. These animals represented a significant step toward the mammalian condition. Their legs began to shift from a sprawling posture to a more upright stance, tucking underneath the body rather than extending out to the sides. This change, while gradual and incomplete in many species, would eventually allow their descendants to move with greater stamina and efficiency. The therapsids diversified into an array of ecological roles. Some became formidable predators. Others evolved into specialized herbivores, and still others took to burrowing underground. The Dicynodonts, whose name means two dog teeth, emerged as particularly successful herbivores. These animals ranged from small burrowers to large herd animals like Placerius, which resembled a pig-sized, tusked creature that roamed in groups across the late Permian landscape. The most significant group of therapsids for the mammalian story were the cynodonts, whose name means dog teeth. These animals appeared in the late Permian and would prove to be direct ancestors of all modern mammals. Thrinaxodon, a dog-sized carnivorous cynodont from what is now South Africa, exemplifies the transitional nature of these animals. Its teeth had begun to differentiate into specialized types for cutting, biting, and chewing. Evidence suggests Thrinaxodon possessed whiskers, indicating a sophisticated sense of touch. Fossils of another cynodont called Triracodon have been found in multi-chambered burrows, with as many as 20 skeletons discovered together, apparently drowned by a flash flood. This suggests these animals engaged in complex social behaviors, living in communal groups rather than as solitary individuals. Then catastrophe struck. The Earth experienced the most devastating extinction event in its history. The cause remains debated, with evidence pointing to massive volcanic eruptions, possibly compounded by an asteroid impact. Whatever the trigger, the results were apocalyptic. Marine ecosystems collapsed. On land, forests withered and disappeared. The dominant predators that had ruled the late Permian vanished completely. Yet some synapsids survived. Dysonants like Lystrosaurus and Placerius endured. Most importantly, the cynodonts made it through the bottleneck, and they were the only mammal-like reptiles to survive into the Jurassic and coexist with early dinosaurs, carrying with them the genetic legacy that would eventually flower into the full mammalian condition. The world these survivors inherited was dramatically different from the one their ancestors had known. As ecosystems slowly recovered over millions of years, a previously obscure group of reptiles called archosaurs began to diversify and spread. This group would eventually give rise to crocodiles, pterosaurs, and dinosaurs. By the Middle Triassic, these archosaurs had begun to dominate the large-bodied terrestrial niches. The cynodonts, which had once included sizable predators, found themselves increasingly marginalized. This ecological pressure may have been the crucible in which true mammals were forged. As archosaurs claimed the roles of large diurnal predators and herbivores, the cynodonts adapted by going in the opposite direction. They became smaller, more specialized, and shifted to nocturnal lifestyles. This retreat into the night and into smaller body sizes demanded new adaptations. Active hunting in darkness required acute hearing and an excellent sense of smell. Maintaining a high metabolism in a small body required efficient food processing and better insulation. This period has been called the nocturnal bottleneck, a hypothesis suggesting that the constraint of living at night while dinosaurs ruled the day drove many distinctively mammalian traits. Around 225 to 210 million years ago, in the late Triassic, the first animals that can be called true mammals appeared. The line between mammal and non-mammal is not sharp, but rather represents a threshold where enough distinctly mammalian features had accumulated. Morganucodon watsoni, first discovered in Welsh caves but later found in China, India, North America, South Africa, and Western Europe, represents this threshold. This shrew-sized creature, only about 10 centimeters long excluding its tail, had teeth that were replaced just once in life rather than continuously, moving toward the mammalian pattern of baby teeth and adult teeth. It almost certainly laid eggs, as the first mammals were egg layers, but it likely already possessed fur and may have produced some form of milk to nourish its young. The jaw joint evolved, eventually freeing some bones to migrate into the middle ear, a transformation that would give mammals their exceptional hearing. Other innovations emerged: a bony palate separating breathing from eating, increasingly complex teeth for efficient food processing, and specialized glands for nourishing young. These changes did not happen all at once, nor did they happen in lockstep. Different mammalian features appeared at different times and in different lineages, with some groups developing certain traits while lacking others. The next chapter will explore how these key mammalian traits evolved and what advantages they provided to early mammals pursuing their nocturnal existence. What drove this transformation from sprawling, reptilian synapsids to the first true mammals was not a single environmental pressure, but a complex interplay of competition, opportunity, and accident. The Great Extinction wiped out most synapsid diversity, but left survivors that were perhaps predisposed to certain adaptations. The rise of dinosaurs and other archosaurs forced the remaining synapsids into ecological corners where small size, nocturnal habits, and metabolic efficiency became advantageous. Yet within those constraints, the ancestors of mammals found not limitation but possibility. And in the shadow of the dinosaurs, they were quietly assembling the toolkit that would one day allow their descendants to inherit the earth. Chapter 2. The Architecture of Mammalness. A shrew-sized creature dies in a Jurassic forest, its body settling into fine-grained sediment that will, over millions of years, compress into stone. By extraordinary chance, the sediment preserves not just bone, but the delicate impressions of soft tissue. When paleontologists split the rock 164 million years later, they find something that should not have survived, the unmistakable imprint of fur. This is the evidence that anchors our understanding of when mammals became recognizably mammalian. But the story of how that transformation occurred requires reading clues scattered across far older stones. The evidence for fur pushes surprisingly far back in time. When scientists in the 1950s examined the skulls of cynodonts, they noticed numerous small holes, or foramina, in the maxillae and premaxillae bones at the front of the upper jaw. These passages resembled those in modern mammals that supply blood vessels and nerves to vibrissae, the specialized sensory hairs we commonly call whiskers. The logic seemed sound. Whiskers require elaborate nerve supply, and these holes suggested such supply existed. Therefore, cynodonts must have had whiskers, and if they had whiskers, they probably had fur covering the rest of their bodies as well. This interpretation was soon challenged. Modern lizards such as Tupinambus possess foramina nearly identical to those found in cynodonts, yet lizards have scales, not fur. The holes alone proved nothing definitive about hair. The debate might have remained unresolved except for the discovery of remarkable coprolites from the Permian period, the fossilized feces of predators that fed on dicynodonts. Within these ancient droppings, paleontologists identified elongated hollow structures consistent with hair shafts. This pushed the origin of fur back to at least 252 million years ago, suggesting that even some of the earlier therapsids, well before the emergence of true mammals, had already evolved this insulating covering. The first direct, indisputable evidence comes from Middle Jurassic fossils preserving clear impressions of dense fur. These discoveries confirm that by 164 million years ago, mammalian fur had achieved its modern characteristics, with dense underfur providing insulation and possibly longer guard hairs offering protection from water and physical damage. The purpose of fur extends beyond simple warmth. In small-bodied animals, especially those active at night, the problem of heat loss becomes acute. Surface area increases relative to body volume as animals shrink, meaning smaller creatures lose heat faster. Fur creates a boundary layer of still air next to the skin, dramatically reducing heat loss through convection. But fur also serves another crucial function in thermoregulation. It can trap heat during cold periods and, when moistened with sweat or other secretions, facilitate evaporative cooling during heat stress. This dual function would have been essential for early mammals pursuing their nocturnal insectivorous lifestyle, hunting actively during cool nights and retreating to burrows during warmer days. The evolution of teeth in the mammalian lineage represents one of the most thoroughly documented transformations in the fossil record. Teeth are exceptionally durable, often outlasting all other parts of the skeleton, and their complex shapes record vast amounts of functional information. The earliest synapsids had relatively simple, uniform teeth that continuously grew throughout life, a condition called polyphyodonty. These teeth could grasp and pierce, but could not efficiently process food through chewing. Cynodonts began to change this pattern. Their teeth differentiated into distinct types: incisors at the front for nipping and cutting, prominent canines for puncturing, and a series of postcanine teeth toward the back of the jaw. Some of these rare teeth developed multiple cusps arranged in specific patterns, allowing them to interlock when the jaws closed. This differentiation of tooth types, called heterodonty, required a corresponding change in jaw mechanics. Reptiles typically possess many points of articulation in their jaws and often have teeth that continue replacing throughout life. Mammals, by contrast, have fewer jaw bones and typically replace their teeth only once, going from deciduous baby teeth to permanent adult teeth. This pattern, known as diffyodonty, appears in the earliest true mammals. The transition from continuous replacement to limited replacement was not merely a curiosity of dental development. It was intimately connected to the precision chewing that mammals would perfect. When teeth continuously replace throughout life, the chewing surfaces cannot develop precise matching patterns between upper and lower jaws. Each tooth is slightly different from its predecessor, making fine-tuned occlusion impossible. By limiting tooth replacement to a single juvenile event, mammals ensure that their adult teeth can wear in together, developing exact matching surfaces that allow molars in the upper and lower jaws to meet in precise alignment. This precision occlusion, where the cusps of upper molars fit into the valleys of lower molars and vice versa, permits a grinding and crushing action far more efficient than anything possible with simpler teeth. The cynodonts and early mammals developed increasingly complex molar patterns. The Morganuconatids possessed molars with cusps arranged in a triangle, with upper and lower molars interlocking to slice food into pieces as the jaws closed. Later groups refined this into the tribosphenic molar, a remarkably sophisticated design that appeared in both marsupial and placental lineages. The tribosphenic molar has a triangular arrangement of cusps on the upper molar and a basin surrounded by cusps on the lower molar. As the jaws close, the upper cusps shear past the lower cusps and then crush food by pressing into the lower basin. This design allows simultaneous cutting and grinding in a single chewing stroke, dramatically increasing the efficiency of food processing. Why does this matter? Chewing breaks food into smaller particles, which increases the surface area available for digestive enzymes to act upon. This accelerates digestion and allows more complete extraction of nutrients from food. For small, active animals with high metabolic rates, this efficiency is essential. A cynodont or early mammal that could extract more energy from each meal could afford to spend less time eating and more time on other activities, including hunting, avoiding predators, or caring for young. But chewing creates a problem. You cannot breathe easily through your nose while your mouth is full of food being ground by powerful jaws. The solution emerged in the form of the secondary palate, a shelf of bone that separates the nasal passages from the mouth cavity. Early synapsids lacked this structure entirely. Air passed through the nostrils into a space that was also the forward part of the mouth. Cynodonts began developing a partial secondary palate, and by the time of the first true mammals, this bony shelf extended all the way to the back of the mouth, completely separating breathing from eating. The secondary palate allowed proto-mammals and mammals to breathe continuously while processing food, maintaining oxygen supply to their active tissues even during the mechanical work of chewing. This innovation directly supported the possibility of sustained activity and higher metabolic rates. The palate also provided a surface for the tongue to manipulate food during chewing, but both functions reinforced the same evolutionary trajectory toward more efficient food processing and higher energy throughput. The transformation of the jaw itself ranks among the most remarkable transitions in vertebrate evolution. In early synapsids, the jaw joint formed where the articular bone of the lower jaw met the quadrate bone of the skull. This is the ancestral condition for all tetrapods, inherited from their fish ancestors. Multiple bones made up the lower jaw, with the dentary being just one element, albeit an increasingly important one. As synapsids evolved, the dentary bone gradually enlarged, taking on more of the function of supporting the teeth and anchoring jaw muscles. By the time of the advanced synodonts, the dentary had become the dominant bone of the lower jaw. Yet the ancient jaw joint between articular and quadrate remained, even as a new articulation began to form between the dentary and the squamosal bone of the skull. For a period, some mammalian ancestors possessed a dual jaw joint, with both the old articulation and the new functioning simultaneously. This dual arrangement appears clearly preserved in transitional forms, demonstrating that evolution does not always proceed through clean replacements, but sometimes works with redundant systems that gradually shift function. Eventually, the dentary squamosal joint took over completely. The articular and quadrate bones, no longer needed for jaw support, became freed for other functions. In a transformation that still astonishes anyone who studies it carefully, these bones migrated inward and became part of the middle ear. The quadrate became the incus, and the articular became the malleus, two of the three tiny bones that conduct sound from the eardrum to the inner ear in mammals. The third bone, the stapes, was already part of the ear in earlier vertebrates, but it now formed a chain with two additional elements, creating a more efficient sound transmission system. This transformation can be traced in exquisite detail through the fossil record. Hadrocodium, from the early Jurassic, shows a jaw structure suggesting the ear had already moved fully into the cranium, encased in solid bone, rather than attached to the jaw through a trough of bone. This architectural shift insulated the ear from vibrations caused by chewing, allowing mammals to hear while eating without the sound of their own jaws overwhelming external sounds. This transformation can be traced in exquisite detail through the fossil record. Hadrocodium, from the early Jurassic, shows a jaw structure suggesting the ear had already moved fully into the cranium, encased in solid bone, rather than attached to the jaw through a trough of bone. This architectural shift insulated the ear from vibrations caused by chewing, allowing mammals to hear while eating without the sound of their own jaws overwhelming external sounds. The advantage of this elaborate middle ear system is profound. The chain of three tiny bones acts as a lever system, amplifying the relatively weak vibrations of the eardrum and transmitting them efficiently to the fluid-filled inner ear. This arrangement gives mammals exceptional hearing sensitivity, particularly for high-frequency sounds. For small nocturnal animals hunting insects in darkness, the ability to hear the flutter of moth wings or the rustle of a beetle in leaf litter could mean the difference between feeding and starving. The evolution of mammary glands presents a different kind of puzzle because, unlike bones and teeth, these structures leave no direct fossil evidence. They are modified sweat glands that produce a nutrient-rich secretion we call milk. In modern mammals, the young depend entirely on milk for their initial nutrition, sometimes for months or even years. This dependency requires substantial maternal investment, but it provides offspring with a reliable, high-quality food source perfectly tailored to their needs. The original function of the secretions from these proto-mammary glands may have been to keep eggs moist. Modified sweat glands might initially have helped prevent eggs from drying out, while providing some antimicrobial protection, as many mammalian skin secretions have defensive properties against bacteria and fungi. As the nutritional content of these secretions increased, perhaps through selection for offspring that could supplement their diet by consuming the secretions, the system gradually shifted toward one where the secretions themselves became the primary food source for very young animals. Eventually, this led to the modern mammalian pattern, where milk is essential for survival of offspring. The connection between mammary glands and the shift from polyphyodonty to diphyodonty becomes apparent when you consider the needs of nursing young. A mammal infant without teeth or with only temporary baby teeth can subsist entirely on milk. This allows for an extended period of rapid growth and brain development without the need for the young animal to hunt or forage for itself. The adult teeth erupt only after weaning, when the animal is ready to process solid food efficiently. This timing would be impossible in a system where teeth continuously replace throughout life, as there would be no stable period where the young animal could have a mouth properly configured for nursing but not yet ready for adult feeding. The loss of genes for producing egg yolk proteins provides molecular evidence for this transition. Viviparous placental mammals, which give birth to well-developed live young rather than laying eggs, have lost functional VIT genes that code for vitellogenin, the primary egg yolk protein. These genes decayed into non-functional pseudogenes as lactation and placentation made egg yolk dispensable. The marsupials, which give birth to very underdeveloped young that complete most of their development while attached to a nipple, show an intermediate condition. This molecular evidence confirms that the evolution of milk production was linked to changes in reproductive strategy across the mammalian family tree. These traits, fur for insulation, complex teeth for efficient chewing, a secondary palate for simultaneous breathing and eating, sensitive ears for hunting in darkness, and milk for nourishing young, did not evolve in isolation. Each innovation made others more valuable. Fur enabled sustained activity in cool conditions, which increased the energy demands that efficient chewing could meet. Better food processing supported the metabolic costs of producing milk and maintaining large energy-hungry brains. Acute hearing allowed nocturnal hunting that avoided daytime predators. Together, these features created something fundamentally new, a way of being a vertebrate that opened possibilities no earlier synapsid could have exploited, and set the stage for the ecological diversification that would follow once opportunity allowed. Chapter 3. Mesozoic Mammal Diversification. Night is falling in the early Jurassic, 185 million years ago, and the Cyanthitherium is tending to her newly hatched brood. Heavy rains pummel the bank above her den as she looks over her dozens of tiny young, each no larger than a human thumb. The burrow, carved into soft earth near a river in what will one day be Arizona, offers shelter from the storm. At some point, without warning, the sodden bank collapses, entombing the hatchlings and their mother in mud. The catastrophe preserves them perfectly. When paleontologists uncover this scene millions of years later, they will find the mother's skeleton curled protectively around 38 juveniles, a snapshot of mammaliaform parental care frozen in stone. For most of the 20th century, paleontologists believed they understood the role of mammals during the Mesozoic era. These tiny creatures, the thinking went, lived unremarkable lives as small insectivores, scurrying through the undergrowth, while dinosaurs dominated every significant ecological role. They were bit players in a dinosaur drama, waiting patiently for the asteroid that would end their reptilian overlords and finally give mammals their chance. This comfortable narrative collapsed under the weight of new fossil discoveries beginning in the mid-1990s. The revolution began in northeastern China in a region called Liaoning Province. Here, layers of volcanic ash and sediment had buried entire ecosystems during the Jurassic and Cretaceous periods, preserving not just bones, but soft tissues, fur, feathers, and stomach contents. Paleontologists working these deposits have described more than 24 fossil mammaliaform species from deposits spanning roughly 40 million years, and many of them looked and behaved nothing like the mousy insectivores of scientific tradition. Consider Castor-Acuta, whose name means beaver tail. When its nearly complete skeleton was first described in 2006, it shattered expectations. This mammaliaform lived approximately 164 million years ago during the Middle Jurassic and stretched about 43 centimeters from nose to tail tip, with the flattened tail itself accounting for 13 centimeters. The skeleton revealed adaptations for swimming that were unmistakable. The tailbones were flattened, and soft tissue impressions showed webbing between the toes of the hind feet. The forelimbs were robust and powerful, suitable for paddling or digging. The teeth told another story entirely. The first two molars had cusps arranged in a straight line rather than the triangular pattern seen in most mammalia forms. These blade-like teeth curved backward, perfect for gripping slippery prey. The combination of swimming adaptations and specialized dentition pointed to a single conclusion: Castor acuta hunted fish. This was not supposed to be possible. Until its discovery, the earliest known semi-aquatic mammalia forms came from the Eocene epoch, roughly 110 million years later. If Castor acuta swam through Jurassic rivers, Volaticotherium took to the air, or at least the spaces between trees. This mammalia form, discovered in the same region and time period, weighed a mere 70 grams and measured 12 to 14 centimeters in length. Its skeleton preserved clear evidence of a patagium, the membranous wing formed by skin stretched between elongated limbs. The structure closely resembled that of modern gliding mammals, such as flying squirrels, though Volaticotherium was not closely related to any living mammal group. The presence of gliding adaptations this early in mammalian history indicates that the three-dimensional forest environment offered opportunities that at least some mammalia forms exploited. A related form called Velavolodon possessed similar gliding adaptations. Meanwhile, Agilodocodon developed powerful grasping digits and a flexible backbone suited for climbing through tree branches, suggesting yet another approach to arboreal life. These discoveries demonstrated that multiple independent lineages of mammalia forms experimented with life above the ground, each finding its own solution to the challenges of the canopy. While some mammalia forms swam and others glided, still others went underground. Frutifossor, described from late Jurassic deposits, showed clear adaptations for digging. Another digger, Docofossor, resembled modern moles in its body proportions and powerful forelimbs equipped with broad claws designed for excavating soil. These burrowing mammalia forms likely pursued insects and other invertebrates underground, exploiting a resource base that surface-dwelling hunters could not easily access. The dietary specializations of Mesozoic mammalia forms extended far beyond simple insectivory. Analysis of tooth structure and jaw mechanics reveals that different groups processed radically different foods. The Docodonts, an early diverging mammalia form group, showed remarkable dietary diversity among their various species. Some possessed robust jaws capable of crushing the hard exoskeletons of large beetles. Others had gracile jaws and delicate teeth, suited only for soft-bodied prey like moth larvae or earthworms. Still other docodonts evolved peculiarly mobile jaws and specialized molars that could slice and grind plant material, making them among the first herbivorous mammalia forms. The Haramiyidans represent another early branching group that adopted plant eating. Their molars developed unique grinding surfaces, and the jaw joint permitted unusual side-to-side movement during chewing, allowing efficient processing of tough plant fibers. These herbivorous specialists occupied niches that, in other ecosystems, might have been filled by small reptiles or insects. The most dramatic evidence of mammaliaform dietary diversity comes from a spectacular fossil discovered in China's early Cretaceous deposits. Repenomamus giganticus lived approximately 130 million years ago and stretched over a meter in length. It weighed an estimated 12 to 14 kilograms, making it by far the largest mammalia form known from the Mesozoic. But size alone would not have made Repenomamus remarkable. What shocked paleontologists was a second specimen of a related species, Repenomamus robustus, found with the bones of a juvenile psittacosaur preserved in its rib cage. The baby dinosaur's bones showed evidence of having been broken and partially digested, proving this was a genuine meal rather than accidental association of fossils. Suddenly, the image of Mesozoic mammals as timid creatures cowering from dinosaurs required revision. At least one mammaliaform species had turned the tables, preying on dinosaurs rather than hiding from them. The multituberculatids deserve special attention as perhaps the most successful group of non-therian mammaliaforms. They first appeared in the Jurassic period and would persist for approximately 130 million years, not going extinct until roughly 35 million years ago. To put this in perspective, the entire span of time from the first recognizable primates to modern humans encompasses only about 65 million years. The multituberculatates outlasted most dinosaur lineages and survived longer than any other non-therian mammaliaform group. Their success came from dental innovation. Multituberculatates possessed enlarged front teeth that worked like chisels, similar to those of modern rodents, and complex molars with multiple rows of cusps that could grind plant material. Some multituberculatates also developed specialized premolars with blade-like edges for slicing. This combination of shearing and grinding capability allowed them to process a wide variety of plant foods, from seeds to leaves to bark. These varied mammalia forms shared their world with other early mammalian lineages. The Eutriconodonts, named for their distinctive three-cusped teeth, included forms ranging from small insectivores to larger predators. The Spalacotherids developed specialized teeth that suggest they too consumed insects and other small prey. Meanwhile, the earliest members of the three lineages that survive today, monotremes, marsupials, and placentals, were present by the Cretaceous, though they remained relatively rare compared to multituberculates and other groups that would later become extinct. Mammalia forms displayed astonishing diversity. They exploited resources in the water, in the air, underground, in trees, and on the ground. They ate insects, fish, plants, and occasionally even baby dinosaurs. They employed different locomotor strategies, from swimming to gliding to digging. They varied in their sensory emphasis, with some relying heavily on hearing, others on smell, and still others on touch through specialized whiskers. The evolutionary experimentation of the Mesozoic produced numerous dead-end lineages. Gliding evolved independently at least twice. Swimming adaptations arose separately in different groups. Herbivory appeared in multiple unrelated mammaliaform clades. Most of the mammaliaform diversity of the Jurassic and Cretaceous did not survive to the present. The multituberculates, docodonts, haramiodonts, eutriconodonts, and numerous other groups all went extinct, leaving only the monotremes, marsupials, and placentals to carry forward. Why some lineages persisted while others perished would only become clear when the world that dinosaurs and mammaliaforms shared came to a sudden and catastrophic end. Chapter 4. When Giants Fell. The history of mammalian evolution cannot be understood as a simple story of gradual improvement and expansion. Instead, it is punctuated by catastrophic disruptions that repeatedly reset the biological deck, eliminating dominant groups and creating opportunities for survivors to radiate into vacant ecological space. Three major extinction events shaped the trajectory of mammalian evolution, each leaving an indelible mark on which lineages would thrive and which would vanish from Earth forever. The Permian-Triassic extinction event was not a single apocalyptic moment, but rather a prolonged crisis unfolding in waves. The extinction occurred in multiple pulses, possibly spanning hundreds of thousands of years around 252 million years ago. Evidence from marine sediments shows repeated episodes of ocean acidification and anoxia, periods when oxygen levels dropped so low that most marine life suffocated. On land, the crisis manifested differently but no less devastatingly. The primary suspect for triggering this cascade of death is the Siberian Traps, a massive volcanic eruption that covered an area larger than Europe with lava. Unlike the brief violence of an asteroid impact, these eruptions persisted for perhaps 1 million years, pouring megatons of carbon dioxide, sulfur dioxide, and other gases into the atmosphere. The carbon dioxide would have caused rapid global warming, while sulfur dioxide would have produced acid rain. Researchers have suggested that the warming became so extreme that it released vast quantities of methane frozen in ocean sediments, amplifying the greenhouse effect to lethal levels. The synapsids, which had dominated terrestrial ecosystems for tens of millions of years, were devastated. The large Gorgonopsian predators vanished completely. Most dicynodont lineages died out. The therapsid groups that had filled numerous ecological roles, from burrowers to apex predators to large herbivores, were pruned back to a handful of surviving lineages. What emerged from the bottleneck was not a representative sampling of Permian diversity, but rather a biased subset. Those species that happened to possess characteristics that allowed survival under the new conditions. Lystrosaurus, the pig-sized dicynont, became extraordinarily abundant in the early Triassic, at some localities making up 95% of vertebrate fossils. This dominance by a single genus suggests an ecosystem severely impoverished of diversity, where one generalist species could spread unchecked across a simplified landscape. The cynodont lineages that survived were mostly smaller-bodied forms, already adapted to burrowing or other strategies that might have buffered them against the environmental chaos. The recovery from the Permian-Triassic extinction took an extraordinarily long time by the standards of mass extinctions. Ecosystems and food chains remained unstable for approximately 30 million years before anything approaching normal complexity returned. During this prolonged recovery, the archosaurs, which had been minor players in Permian ecosystems, diversified and spread. By the time ecosystems had stabilized, archosaurs, rather than synapsids, occupied most large-bodied terrestrial niches. The synapsids never regained their former dominance. Instead, the surviving cynodont lineages adapted to life in the margins, pursuing strategies based on small size, nocturnal activity, and metabolic efficiency. A second extinction event occurred at the end of the Triassic period, roughly 201 million years ago. While smaller in scale than the Permian-Triassic catastrophe, this event nonetheless reshaped terrestrial ecosystems and determined which lineages would dominate the coming Jurassic period. The causes remain debated, with evidence pointing to volcanic activity in the Central Atlantic magmatic province, massive eruptions associated with the breakup of Pangaea that released carbon dioxide and sulfur compounds into the atmosphere. Many archosaur lineages died out during this event, but crucially, the dinosaurs survived and subsequently radiated into the dominant large animals of the Jurassic. The timing of this turnover appears significant. Before the extinction, dinosaurs shared terrestrial environments with various other archosaur groups, including large predatory Rawasuchians and armored herbivorous Eotosaurs. After the extinction, these competitors had vanished, leaving dinosaurs to fill the vacated ecological roles. Within a few million years, dinosaurs had diversified into the familiar groups that would dominate for the next 135 million years. Among synapsids, some of the remaining non-mammalian synodonts went extinct, narrowing the diversity of the mammalian stem group. However, the true mammals, which had only recently emerged, survived this event. Their small size and specialized nocturnal ecology likely provided some protection. The pattern emerging across these extinctions became clear. Large-bodied specialists suffered disproportionately, while small-bodied generalists with flexible behavior tended to persist. Between the Triassic-Jurassic event and the next great catastrophe stretched 135 million years, a vast span during which dinosaurs ruled and mammals remained largely relegated to small body sizes and nocturnal niches. Then came the third great extinction. 66 million years ago, at the boundary between the Cretaceous and Paleogene periods, a massive asteroid, perhaps 12 kilometers in diameter, struck what is now the Yucatan Peninsula in Mexico, creating the Chicxulub crater. The immediate effects were apocalyptic. The explosion released energy equivalent to billions of nuclear weapons, triggering earthquakes, tsunamis, and global firestorms. Dust and debris blasted into the stratosphere, blocking sunlight and causing temperatures to plummet. Photosynthesis shut down across much of the planet. Plants died. Herbivores starved. Predators found no prey. When the dust finally settled, over 70% of all species on Earth had been wiped out, including every non-avian dinosaur. The question of which mammals survived and why has occupied researchers for decades. Intriguingly, some successful Mesozoic mammalian lineages did not make it through. 5 of the 15 mammal families known from the end of the Cretaceous became extinct during or immediately after the impact. The early members of placental and marsupial lineages made it through, but many other groups, including various mammaliaform lineages that had experimented with swimming, gliding, and specialized diets, vanished entirely. Size appears to have been crucial. Smaller animals generally fared better than larger ones, possibly because smaller animals require less food, can find shelter more easily, and can survive on scattered or marginal resources. The largest mammals of the latest Cretaceous were in the size range of modern badgers. After the extinction, the immediate survivors were even smaller, mostly ranging from mouse to rat-sized. Diet likely mattered as well. Species dependent on fresh plant material would have suffered as vegetation died back. Specialized predators of dinosaurs obviously had no future. The survivors were probably generalists, animals that could switch between different food sources as circumstances demanded. Insectivores and omnivores had advantages, as insects can survive on detritus and carrion, and omnivores can exploit whatever happens to be available. The ability to shelter underground or in water may have provided critical protection from the initial heat pulse and subsequent environmental chaos. The recovery of mammalian diversity after the extinction followed a remarkably rapid trajectory compared to the sluggish recovery from the Permian-Triassic event. Work by Tyler Lyson and colleagues analyzing fossils from Colorado provided an unusually complete record of the first million years after the impact. The data reveal that mammalian taxonomic richness doubled within the first 100,000 years. This means that the number of distinct mammal groups present in the ecosystem increased by a factor of 2 in a geological blink of an eye. Size recovery occurred on a similar timescale. In the immediate aftermath of the extinction, the largest mammals weighed around half a kilogram. Within 100,000 years, mammals had returned to the maximum size seen before the extinction, approximately 7 kilograms. By 300,000 years after the impact, the largest mammals had increased to 15 kilograms, doubling the pre-extinction maximum. This pattern shows that ecological opportunity, not physiological limitation, had constrained mammal size during the Mesozoic. The key to understanding this rapid diversification lies not just in the removal of dinosaurian competition, but in the parallel evolution of flowering plants and their associated insect pollinators. The Cretaceous had seen an explosion of angiosperm diversity, and many of these plant lineages survived the extinction event. As ecosystems recovered, plants underwent their own radiation, creating new food sources that mammals could exploit. The fossil record from Colorado reveals 3 distinct warming intervals in the first million years after the extinction, each associated with pulses of evolutionary change in both plants and mammals. These warming events may have been triggered by ongoing volcanic activity from the Deccan Traps in India. Though initially suspected as the primary cause of the extinction itself, current evidence strongly favors the asteroid impact as the main killer. However, the volcanic activity continued after the impact, and the carbon dioxide it released caused temperature fluctuations that influenced the recovery process. During each warming interval, forest ecosystems re-established themselves more completely. Forests provided new 3-dimensional habitats with abundant food resources in the form of leaves, fruits, seeds, and insects. Mammals responded by diversifying and by increasing in size. Approximately 700,000 years after the extinction, the bean family of flowering plants, Fabaceae, appeared in the fossil record. These nitrogen-fixing plants produce protein-rich seeds highly nutritious for herbivores. Their arrival coincided with another surge in maximum mammal body size, reaching 50 kilograms, suggesting a direct link between plant innovation and mammalian evolution. The marsupials and placentals both survived the extinction and both radiated afterward, but they did so in different parts of the world with different outcomes. Marsupials achieved their greatest diversity in South America and Australia, continents that became isolated during the Cenozoic. Placentals dominated in the Northern Hemisphere and Africa. This geographic separation allowed both groups to diversify independently, evolving parallel solutions to similar ecological challenges. Molecular evidence later confirmed that modern placental orders arose after the extinction, supporting what the fossil record had long suggested about the timing and pace of post-catastrophe diversification. These extinction events functioned as evolutionary filters, not random culling. Certain characteristics, small body size, dietary flexibility, ability to shelter, generalist ecology, repeatedly predicted survival. The groups that went extinct were often specialists, animals finely tuned to specific conditions that the extinctions rendered obsolete. The mammals that emerged from these crucibles were not necessarily superior in any absolute sense. They were simply better suited to chaos and to seizing the opportunities that followed, opportunities that would allow them to diversify into forms that would have seemed impossible during the reign of the dinosaurs.
Chapter 5: Mammalian Ascendancy. The world that emerged from the ash and darkness of the asteroid impact was a landscape of possibility. With the extinction of non-avian dinosaurs and the opening of countless ecological roles, the surviving mammals stood at the threshold of an evolutionary explosion that would unfold with astonishing speed. Yet the story of mammalian dominance is not simply one of survivors expanding into vacated spaces. The radiation that followed was far more complex, driven by the emergence of entirely new ecological opportunities that had not existed even when dinosaurs thrived. The Paleocene epoch, spanning from 66 to 56 million years ago, was a time of experimentation and ecological pioneering. Among the earliest mammals to exploit the post-extinction world were the condylarths, a loose grouping of primitive hoofed mammals that would proliferate during this period. Peripticus exemplifies this early phase of mammalian expansion. This creature, roughly the size of a medium to large dog and weighing around 20 kilograms, lived in what is now the southwestern United States between 63 and 62 million years ago. Its robust teeth were adapted for processing tough plant material, roots and stems that required significant grinding force. Its stout skeleton was well suited for terrestrial movement through forest vegetation, navigating fallen logs and dense undergrowth.
Peripticus represented something genuinely new in mammalian evolution, a moderately large terrestrial herbivore. During the Mesozoic, mammals had been constrained to small body sizes, and most were insectivores or omnivores. The condylarths pioneered the exploitation of abundant plant resources, with bodies large enough to process quantities of low-quality vegetation. They were not merely filling the niche of hadrosaurs or ceratopsians. Those dinosaurs had been far larger and possessed different digestive strategies. Instead, condylarths created a new ecological role, that of medium-sized mammalian herbivore adapted to forest environments that were themselves undergoing rapid evolution. Other early Paleocene mammals explored different ecological possibilities. Arctocyon, appearing within the first million years after the extinction, evolved powerful jaws and blunt crushing teeth suited for processing hard foods like nuts and tough invertebrates. This bear-sized omnivore represents one of the earliest large predatory mammals, though it likely scavenged as much as it hunted. Meanwhile, smaller forms like Prototungulatum, weighing perhaps half a kilogram, possessed teeth suggesting it fed on insects and fruits. These early radiations produced mammals occupying niches from tiny insectivores to bear-sized omnivores within just a few million years. The speed of early mammalian diversification defies expectations based on typical rates of evolution. The 10 mammal families that survived the extinction expanded to 78 families by the early Eocene, roughly 10 million years later. This represents nearly an 8-fold increase in family-level diversity in a span that amounts to a brief moment in geological time. Simultaneously, the number of mammalian genera exploded from around 40 to over 200. The pattern fits the classic definition of adaptive radiation, where a lineage rapidly diversifies to fill multiple ecological niches. The Middle Eocene, approximately 45 million years ago, marks a pivotal moment in mammalian history. By this time, all the major groups of mammals found in the modern world had come into existence. This does not mean they looked like their modern descendants. The early representatives of these lineages were often quite different from living forms. But the fundamental branching pattern of the mammalian family tree was established. Primates, rodents, bats, carnivores, whales, elephants, horses, and all the other major orders had emerged and begun their own separate evolutionary trajectories. The Eocene also saw mammals achieving body sizes that began to approach those of large dinosaurs, though still far smaller than the largest dinosaurian herbivores. The climate during much of the Eocene was warmer and more equable than today, with forests extending to high latitudes and supporting lush vegetation. These conditions permitted the evolution of browsing mammals that could process large quantities of plant material. The trend toward larger body size continued through the Oligocene and Miocene epochs. By the Oligocene, some 26 to 23 million years ago, truly gigantic mammals had appeared. Indricotherium, a hornless relative of modern rhinoceroses, stood 5.4 meters tall at the shoulder and may have weighed as much as 20 tons, making it comparable in mass to some of the largest dinosaurian herbivores, though its body proportions were entirely different. Unlike the massive sauropods with their long necks held horizontally, Indricotherium carried its neck more vertically, using its height to reach high into trees to feed on leaves. Its limbs were columnar and elephant-like, supporting the enormous mass through bone architecture that distributed weight efficiently. The skull was elongated and relatively small for the body, with simple teeth suited for browsing. Fossil evidence shows these giants ranged from arid environments with scattered trees to more lush forests across Asia, demonstrating that mammals had overcome whatever physiological constraints might have limited their size during the Mesozoic. Brontotherium, from North America during the same period, presented a different solution to herbivorous gigantism. Reaching sizes comparable to modern elephants and weighing several tons, these animals possessed two large blunt horns on their snouts, Y-shaped structures formed from outgrowths of the nasal bones. The function of these peculiar ornaments remains debated. They may have been used in combat between males, with rival bulls clashing head-to-head in contests for mates. The broad, blunt shape would have spread impact forces across a wide area, perhaps preventing serious injury during ritualized combat. Alternatively, the horns might have served defensive purposes against predators like the formidable creodonts that hunted these Oligocene landscapes. Most likely, they functioned primarily in display, allowing individuals to signal fitness and establish dominance hierarchies without the risks of actual combat. Fossil evidence shows considerable variation in horn size between individuals, consistent with a sexually selected trait where larger horns provided mating advantages. Predators evolved in parallel with these herbivores. The creodonts, an extinct group of carnivorous mammals, included forms that filled roles similar to modern wolves, hyenas, and big cats, though they were not closely related to any living carnivore group. Later, the true carnivora diversified into the familiar families we know today. Among the most iconic of extinct predators was Smilodon, the saber-toothed cat, though this genus did not appear until much later in the Pliocene and Pleistocene. However, saber-tooth morphology evolved independently in multiple mammalian lineages throughout the Cenozoic, suggesting that this hunting strategy was repeatedly favored by selection under certain ecological conditions.
Mammals did not simply expand to fill roles vacated by dinosaurs. They innovated, developing new ways of moving, feeding, sensing, and reproducing that had no direct dinosaurian analogs. Bats evolved powered flight, an ability no non-avian dinosaur ever achieved. The earliest bat fossils, appearing by the early Eocene around 52 million years ago, already show fully developed flight capabilities, with elongated finger bones supporting wing membranes, suggesting the transition occurred rapidly once it began. Whales returned to the ocean and eventually evolved into the largest animals ever to exist, far surpassing even the most massive dinosaurs.
Elephants developed trunks, extraordinarily versatile appendages formed from elongated noses and upper lips containing over 40,000 individualized muscles. Early proboscideans like Phosphaterium from Opalocene lacked trunks, having only slightly extended snouts. By the Oligocene, forms like Paleomastodon possessed short trunks. Later groups extended these structures to the remarkable length seen in modern elephants, creating organs capable of precise manipulation, powerful suction, sensitive touch, and even acoustic communication. Horses evolved specialized single-toed limbs optimized for running at high speeds across open grasslands. The earliest horses, like Eohippus from the Eocene, stood only about 30 centimeters tall and possessed 4 toes on their front feet and 3 on their hind feet. Over tens de millones de años, as grasslands spread durante el Miocene, horse lineages gradually enlarged, lengthened their legs, and reduced their toes until only the middle toe remained, creating the single hoof de modern horses capable of sustained high-speed running.
These innovations were not predetermined by mammalian anatomy. They emerged through the interaction of mammalian traits with new ecological opportunities and selection pressures. The placenta, which allows extended gestation and produces more developmentally advanced young than marsupials or monotrinchs can achieve, may have provided placentals with advantages in certain environments. Different reproductive strategies succeeded in different geographic contexts, but the specific paths taken by mammals on different continents would reveal patterns that anatomy alone could never explain. Patterns that would only become visible when scientists learned to read the molecular record hidden in living cells.
Chapter 6: Isolation and Divergence. The 3 great branches of living mammals diverged from one another deep in the Mesozoic, but their evolutionary stories unfolded in dramatically different ways. Each lineage pioneered distinct solutions to the challenges of survival and reproduction, and each would come to dominate different portions of the globe. Understanding how monotremes, marsupials, and placentals went their separate ways requires looking not just at their anatomical differences, but at the geographic accidents that shaped their respective radiations. The monotremes represent the earliest split in the mammalian family tree, diverging from other mammals somewhere in the Jurassic period. The fossil record offers tantalizing but fragmentary glimpses of this ancient lineage. Taenolaphos, discovered in Australia and dated to approximately 120 million years ago, represents one of the earliest known monotremes, confirming that the lineage has deep roots in the Australian region. Today, monotremes consist of only 5 species, the platypus, and 4 species of echidnas. This represents an extraordinarily restricted remnant of what was presumably once a more diverse group. What makes monotremes remarkable is not their diversity, but their retention of characteristics that other mammals abandoned. They still lay eggs, though these eggs are leathery rather than hard-shelled. The female platypus lacks nipples. Instead, milk oozes from mammary patches on her abdomen, and the young lap it up directly from her fur.
The platypus exemplifies the peculiar evolutionary path of monotremes. Its electroreceptors, embedded in the skin of its bill, allow it to detect electrical fields generated by muscle contractions in prey animals.
When diving in murky water with eyes and ears closed, the platypus sweeps its bill back and forth, sensing crayfish and aquatic insects through bioelectric signatures alone. Recent examination revealed another surprise. Platypuses glow under ultraviolet light, their fur fluorescing green due to unusual pigment structures. Even stranger, platypuses possess 5 pairs of sex chromosomes, rather than the 2 found in most mammals, creating a complex genetic system for determining sex that resembles bird chromosomes more than typical mammalian ones. These oddities suggest that monotreme biology still holds secrets yet to be discovered. The split between marsupials and placentals occurred later, but still well before the end of the Cretaceous. The Eutherian-Mesotherian split likely occurred before 160 million years ago, though Sinodelphus at 125 million years ago represents the earliest known Metatherian fossil, and Jeremiah sinensis at 160 million years ago marks the oldest confirmed Eutherian. The fundamental distinction between marsupials and placentals lies in their reproductive strategies. Marsupials have very short gestation periods, typically measured in days or weeks. The young are born extremely underdeveloped, often little more than embryos with barely formed limbs and closed eyes. The newborn must immediately crawl through its mother's fur to reach a teat, usually inside a protective pouch where it attaches and continues developing while nursing continuously. This pattern requires certain anatomical features. Marsupials retain epipubic bones, paired bones extending forward from the pelvis. These bones limit how much the abdomen can expand, which in turn constrains gestation length. By preventing significant abdominal distension, epipubic bones make prolonged internal development of a large fetus impossible. Placentals lost these bones, allowing the abdomen to expand substantially to accommodate a growing fetus over extended gestation periods. These different strategies carry distinct advantages and disadvantages. The marsupial system allows a female to terminate investment in offspring quickly if environmental conditions deteriorate. A nursing female can simply stop feeding a young attached to her teat, freeing resources for her own survival with minimal physiological cost. The placental system commits the female to a longer period of internal gestation, during which terminating the pregnancy is more metabolically expensive. However, placental young are born more developed and capable, potentially requiring less intensive maternal care after birth. Neither system is inherently superior. Each succeeds under different ecological circumstances.
Geographic isolation determined which system would dominate where. During the Cretaceous and early Cenozoic, the continents occupied different positions than today. South America and Antarctica were connected, and Antarctica was not yet positioned over the South Pole. Australia was linked to Antarctica. This arrangement allowed marsupials to spread from North America, where early Metatherians originated, through South America and across Antarctica to Australia. When the continents later separated, marsupials found themselves isolated on southern landmasses, while placentals dominated in the north. In South America, isolated for much of the Cenozoic, marsupials radiated into an astonishing variety of forms. The Borhianoids were carnivorous marsupials that occupied predatory niches. Mayulastes ferox from the early Paleocene of Bolivia was a small predator with strong limbs adapted for pursuit and climbing. Later Borhianoids grew larger, with some reaching the size of bears. Thylacosmolus, appearing in the Miocene, evolved saber teeth convergent with those of placental cats, demonstrating that marsupials could produce predatory forms rivaling those of placentals. South American marsupials also included herbivores and omnivores, though they never achieved the diversity of hoofed placentals that evolved independently on that continent.
Australia told a similar story, but with even more dramatic results. When Australia separated from Antarctica roughly 45 million years ago, it carried marsupials but very few placentals. In the absence of placental competition, Australian marsupials underwent one of the most spectacular adaptive radiations in mammalian history. They evolved forms that eerily parallel placentals found elsewhere.
The thylacine, or Tasmanian tiger, resembled a dog and hunted similarly, despite being a marsupial predator. Marsupial moles burrow through desert sand using powerful forelimbs, just as placental moles burrow through soil. Kangaroos and wallabies, with their bipedal hopping locomotion, have no direct placental equivalent, representing a uniquely marsupial solution to efficient long-distance travel. The Australian radiation produced herbivores ranging from tiny possums to the extinct Diprotodon, a wombat relative the size of a rhinoceros that browsed on vegetation across the continent. Carnivorous marsupials included everything from small insectivores to the Tasmanian devil, a scavenger and predator with bone-crushing jaws. Gliding marsupials evolved independently, with species like the sugar glider developing membranes for gliding between trees, paralleling the gliding squirrels of placental lineages. Yet despite their success in isolation, marsupials faced challenges when continents reconnected. When South America and North America connected via the Isthmus of Panama roughly 3 million years ago, a great exchange of fauna occurred. Placental carnivores from the north, including cats, dogs, and bears, spread south and largely displaced the marsupial predators. Saber-toothed cats moved into territories where Thylacosmilus had once hunted, and the marsupial predators could not compete. Placental herbivores also moved south, including deer, camels, and tapirs, competing with native South American mammals for resources. However, the exchange was not entirely one-sided. Some South American mammals successfully moved north, including armadillos, porcupines, and opossums. The Virginia opossum today ranges well into North America, demonstrating that marsupials could compete with placentals under some circumstances. Ground sloths also invaded North America, though they would later go extinct. The pattern suggests that while placentals held advantages in certain ecological roles, particularly as large predators, marsupials were not universally inferior. The question of why placentals came to dominate globally while marsupials remained largely restricted to Australia and South America has no simple answer. The extended gestation and more developed young of placentals may provide advantages in stable environments, where maternal survival is likely and where competition for resources is intense. The marsupial strategy may excel in unpredictable environments, where flexibility in reproductive investment is crucial, allowing females to abandon failing pregnancies quickly. But these are tendencies, not absolutes. What stands out from examining these three lineages is the profound role of contingency in evolution. Monotremes, isolated in Australasia, survived with their ancient reproductive mode intact but never diversified extensively. Marsupials, given continents to themselves, proved capable of matching placentals in ecological versatility, producing predators, herbivores, and specialists as diverse as any placental fauna. When barriers fell and faunas mixed, competitive outcomes depended on specific ecological contexts rather than any inherent superiority of one reproductive system over another. The map of mammalian diversity reflects not the inevitable triumph of the best design, but rather the accidents of geography, the timing of land bridge connections, and the unpredictable interplay between innovation and isolation. Chapter 7. When Molecules Rewrote the Tree. For nearly 2 centuries after William Buckland announced the first mammal fossils alongside Megalosaurus, paleontologists built their understanding of mammalian relationships on what they could see: bones, teeth, and occasionally soft tissue impressions. The great classifications of the 20th century, culminating in systematic works that organized thousands of species into orders and families, relied almost entirely on morphological comparison. A mammal with hooves was grouped with other hoofed mammals. A mammal with wings went with other flying mammals. Predators with sharp teeth and retractable claws formed their own coherent cluster. This approach had worked remarkably well for centuries, producing classifications that seemed to make intuitive sense and that organized museums, textbooks, and scientific understanding. Then, in the late 1990s and early 2000s, molecular biologists began extracting and sequencing DNA from mammalian tissues. What they found dismantled much of what paleontologists thought they knew about mammalian evolutionary relationships.
The molecular phylogenies, evolutionary trees constructed from comparing genetic sequences rather than anatomical features, told a radically different story. Relationships that had seemed obvious from bones turned out to be illusions created by convergent evolution. Animals that looked utterly different shared recent common ancestors. The comforting edifice of morphology-based classification collapsed under the weight of genetic evidence, forcing a fundamental rethinking of mammalian evolution. Among the most shocking revelations was the fate of pangolins. These scaled mammals, covered in overlapping keratin plates and possessing long sticky tongues for eating ants and termites, had long been classified near the xenarthrans. The resemblance to anteaters seemed too striking to be coincidental. Both groups had abandoned teeth in favor of elongated snouts and specialized tongues. Both fed primarily on ants and termites. Both had powerful claws for tearing open insect nests. Morphological analysis placed them as relatives, suggesting a shared ancestry of insect-eating specialization. The molecular data told a different story entirely. Genetic comparisons showed that pangolins belong not with xenarthrans but within Loriciothria, specifically as relatives of carnivores like dogs and cats. The resemblance to anteaters was pure convergent evolution. Two entirely separate lineages had independently evolved similar solutions to the challenge of eating social insects. Every morphological similarity between pangolins and xenarthrans reflected parallel adaptation to similar diets rather than inheritance from a common ancestor. Bats presented an equally dramatic rearrangement. For decades, the morphological similarities between bats and primates had suggested a close relationship. Both groups have grasping hands with mobile fingers. Both have relatively large brains and complex social behaviors. Some classification schemes grouped bats and primates together, implying shared descent from a common ancestor with these characteristics.
Molecular phylogenetics demolished this grouping. Genetic evidence placed bats firmly within Loriciothera as relatives of carnivores, hoofed mammals, and whales. Primates, meanwhile, belong to Euarchontoglires along with rodents and rabbits. The apparent similarities between bats and primates resulted not from close relationship, but from independent evolution of features useful for life in complex three-dimensional environments. Once again, morphology had been fooled by convergence. The genetic evidence linking whales to even-toed hoofed mammals initially seemed absurd. How could creatures as different as a blue whale and a cow share a recent common ancestor? Yet the molecular signal was unambiguous and consistent across multiple genes. Whales grouped within Artiodactyla, nested within the group rather than as distant relatives. Further analysis revealed that hippos are the closest living relatives of whales, a relationship completely unexpected from anatomy. The merger of these groups created set Artiodactyla, acknowledging that whales are, in evolutionary terms, highly modified artiodactyls that return to the ocean. This finding forced paleontologists to re-examine the fossil record for early whales. When they did, they discovered transitional forms they had previously misinterpreted at Egypt's Wadi el-Hitan, the Valley of the Whales, where dozens of skeletons lie preserved in exquisite detail. Enormous bodies rest in perfect arrangement, with toothy heads connected to gently arching backbones, ribs splayed outward from the trunk. Following the serpentine spine toward the tail, flattened flippers emerge from the shoulders. Then small bones appear near the tapering end, a pelvis and a leg. These walking whales, like Pachycetus and Ambulocetus from roughly 50 million years ago, showed features linking them to artiodactyls, particularly in the structure of their ankle bones. The astragalus, a bone in the ankle, has a distinctive double pulley shape in artiodactyls that early whales shared. This morphological evidence had been overlooked or misinterpreted before molecular data pointed paleontologists toward the connection. Afrotheria emerged as perhaps the most unexpected grouping of all. This clade, whose members evolved in Africa during the continent's long isolation in the Cenozoic, includes elephants, sea cows, hyraxes, aardvarks, elephant shrews, tenrecs, and golden moles.
The sheer diversity of body forms within this group defies comprehension. Elephants weigh several tons and possess trunks and tusks. Golden moles weigh a few grams and burrow through sand. Manatees are aquatic herbivores. Hyraxes look like guinea pigs but are actually relatives of elephants. No morphological analysis had ever suggested these animals formed a natural group. Only molecular data revealed their common African ancestry, exposing how continental drift shaped mammalian evolution. Each isolated landmass fostered its own unique assemblages. Afrotheria in Africa, marsupials in Australia and South America, distinct placental groups in Laurasia. Recently, paleontologists have dug deeper into the fossil record of southern continents. They are finding evidence of advanced mammals far older than any known in the north, perhaps turning traditional biogeographic models upside down. These examples, pangolins among carnivores, whales among artiodactyls, the unlikely Afrotherian assemblage, demolished long-standing classifications. But molecular data did not just reshuffle relationships. It also sparked fierce debate about evolutionary timing itself. Some early molecular studies, using DNA sequence differences to estimate when lineages split, suggested that modern placental orders diverged deep in the Cretaceous period, perhaps 100 million years ago or even earlier. These molecular clock estimates implied that most major mammalian groups coexisted with dinosaurs for tens of millions of years. Paleontologists strongly resisted these conclusions. The oldest unambiguous fossils of most modern mammalian orders date to the Paleocene or Eocene, after the extinction of non-avian dinosaurs. Either the molecular clocks were wrong, overestimating divergence times through incorrect assumptions about rates of evolution, or the relevant fossils had simply not been found. For 2 decades, this debate generated intense disagreement. The timeline of mammalian evolution became one of the most contentious topics in paleontology and evolutionary biology. The computational challenges of analyzing mammalian phylogeny on a large scale compounded the problem. State-of-the-art Bayesian methods for inferring evolutionary timelines require enormous computational power, particularly when analyzing complete genomes rather than just a few genes. For years, such analyses were simply impractical. The breakthrough came with the development of new computational methods that could handle both genomic datasets and thousands of species simultaneously. A team led by researchers at Queen Mary University of London and the University of Bristol developed a fast Bayesian approach that divided the analysis into substeps. First, they simulated evolutionary timelines using complete genomes from 72 mammal species. Then they used these results to guide simulations for the remaining species, incorporating genetic data from nearly 5,000 mammal species in total. This 2-stage process reduced computation time from decades to months, a 100-fold speed-up that not only made the analysis feasible, but also substantially reduced CO2 emissions from computing. The team had to develop strategies to identify poor quality samples and mislabeled data in genetic databases, ensuring their massive analysis rested on solid foundations. The genomic data provided crucial constraints that limited uncertainty. Complete genomes contain vastly more information than individual genes, allowing the statistical methods to reject implausible evolutionary scenarios more effectively. The team also carefully integrated fossil data, using well-dated fossils to calibrate the molecular clock and account for variation in evolutionary rates across different lineages. The results, published in Nature in 2021, settled the long-standing controversy. The ancestors of modern placental mammal groups originated after the Cretaceous-Paleogene extinction, not before it. The analysis confidently rejected an explosive model of diversification deep in the Cretaceous. Instead, crown placentals, the most recent common ancestor of living placental orders and all its descendants, arose in the late Cretaceous, but the unambiguous diversification into distinct modern orders occurred in the Paleocene and Eocene. This timeline aligned with the fossil record, while also explaining the molecular evidence that had suggested earlier dates. What had confused earlier molecular analyses was the distinction between stem lineages and crown groups. The Eutherian lineage itself is ancient, extending back to the Jurassic. Early members of the placental branch within Eutherians existed in the Cretaceous. But these early placentals had not yet split into the modern orders. They were precursors, stem species that had not yet diversified into the specialized groups we recognize today. The crown groups, the modern orders themselves, arose later, after the extinction event cleared ecological space. This resolution demonstrates how integrating different lines of evidence produces more reliable answers than relying on any single source. Fossils alone can miss early divergences if those lineages remained morphologically similar or occupied environments that rarely fossilize. Molecules alone can mislead if evolutionary rates vary in ways not accounted for by the models. But fossils and molecules together, analyzed with sophisticated statistical methods that properly quantify uncertainty, converge on consistent answers.
Yet even with genomic data and powerful computational methods, some relationships remain uncertain. The resolution at the very base of the placental tree, determining which of the four major clades diverged first, remains somewhat ambiguous. Different analyses support different root positions. Afrotheria might be the earliest diverging placental group, or Xenarthra might be, or the root might lie elsewhere entirely. These uncertainties likely reflect genuinely rapid divergence, where multiple lineages split within a brief evolutionary window that is difficult to resolve even with complete genomes. Perhaps the very base of the placental radiation represents an explosion of diversification so compressed in time that no amount of sequence data can fully untangle it. If molecular biology can so thoroughly overturn classifications built on centuries of anatomical study, revealing that pangolins are carnivore relatives, that whales nest within even-toed ungulates, that African golden moles share ancestry with elephants, what other fundamental misunderstandings might still lurk in our attempts to reconstruct deep evolutionary history from fragments that survive?
The evolution of mammals is not a story of inevitable progress, but of survival, adaptation, and extraordinary contingency. From the single opening in a synapsid skull 320 million years ago to the bewildering diversity of mammals today, this journey has been shaped by catastrophic extinctions, geographic isolation, and relentless innovation. Fur, complex teeth, acute hearing, and milk production emerged not through predetermined design, but through the interplay of chance and necessity. The extinction that ended the dinosaurs did not simply hand dominance to mammals waiting in the wings. It created opportunities that survivors seized with astonishing speed, radiating into forms their Mesozoic ancestors could never have achieved. Modern molecular techniques continue to rewrite what we thought we knew, reminding us that evolution's deepest secrets are still being revealed. As mammals ourselves, we are not the culmination of this process, but merely another experiment in an ongoing story whose next chapters remain unwritten. Thank you for listening.